So now we're going to talk about the topic of receptor adaptations. When there's too much stimulation of a receptor on a cell, what happens is that the cell can become desensitized. We call that desensitization. There's a decreased cellular response. There's a couple of general ways that you could think about how this could happen. First possibility is there could be a decreased signal transduction. Something is causing the signal transduction mechanism to not work as well. Another possibility is there could be a decreased number of receptors expressed on the surface, and we call this down regulation. On the other hand, if there is too little stimulation, you can get what's called the opposite, which would be sensitization, which would mean there's an increase in cellular response. And it would really just be the opposite, right? It could be increased in signal transduction, or number two, increased number of receptors expressed on the surface. So what we're going to do is we're going to use the example of a G protein coupled receptor, C to 7 transmembrane spanning domains. Here's the heterotrimeric complex bound GDP to the alpha subunit in the inactive state. Normally we see when we have activation, we have the dissociation of the beta gamma alpha, GTP is bound, and we have the active G protein, which then can go off and have its effect. So if we have the example of too much stimulation, as just an example, of the ligand, too much ligand binding, what will happen in this example of a G protein coupled receptor is that you can start to activate what are called GERCs, G protein coupled receptor kinases. And what these kinases can do is they can phosphorylate the C-terminal part of the receptor. And by doing that, that can cause decreased interactions with the G protein. And that would be very much like the option number one here, where there was a decrease in signal transduction. The other thing that can happen is that when you have the phosphorylation of the receptor, and so we can draw another one in here, and we can put those phosphates in again from the GERC, those can serve as an attachment site for proteins that can bind then to the receptor. These we can just call these arrestin proteins. And when the arrestin protein can bind at a receptor like this, it triggers the receptor to be internalization of the receptor. And there's mechanisms that are involved in this process. And what happens is that this receptor, when it's taken in, can go into the lysosome and eventually the endosome, and you end up having receptor degradation. And so this would be an example of down regulation. And so the idea is that there are different receptors in the G protein coupled receptor family that are more sensitive than others to downregulation, and similarly to upregulation, lack of stimulation. And so this would be an explanation for those observations where you have a decreased response into when there's excess stimulation and increased response when there's not enough stimulation. Keep in mind that these processes are occurring all the time. The receptors are going to the surface, they're being taken in. This is not an all or nothing process. And so when you think about when there's too little stimulation, you would just be having less of this occurring, which would result in the opposite, which would be increased signal transduction and increased number of receptors on the surface.